All right. Okay, it's 11:30. Everybody find a seat, please. Thank you. Okay, so today's advertisement, whatever, is for the Exam 8 session by SOS Western, a group that tries to help students pass their exams while also raising funds for educational development projects in Latin America. So feel free, it's not free, feel free to attend it and make a donation if you can. And hopefully you get some help with for the finals. Okay, so last time we stopped here, if I remember correctly, with the different functions of proteins. And we had learned about how translation works and how proteins are modified after they are being made and how they are being distributed in the cells. And we'll follow up on this distribution on how cells get to know where they have to go within the cell by learning about protein distribution. In bacteria, we've mentioned before that they don't have a real nucleus in the cell, but there's still three different places that a protein can go in the bacterial cell. So it can either stay in the cytosol where it's being made, it can be localized to the membrane of the bacteria, or sometimes proteins are excreted. So excreted proteins in bacteria often have the function of breaking down food sources that are too big to just take up. So they will do their job outside of the cell, and then the cell can take up smaller versions of the food sources that they would like to utilize. In eukaryotes, it's always worse. We have the cytosol, the membrane, we have excreted proteins, the ER, endoplasmic reticulum, mitochondria, nucleus, chloroplast, Golgi, lysozymes, many different ways that the cells has to put many different areas the cell has to put a protein. And then in eukaryotic cells, they're large. So if you have a nerve cell from the nervous system, they are very large. They form those axons where they form those large stretchy things. And sometimes the proteins have to be transported to the front of that section. So there's within the cell, there's structures that can transport a protein inside a cell, which is pretty cool. But so how do the proteins know where to go, or how does the cell know where to put a specific protein? This is usually done by recognizing a amino acid sequence within the protein. So there are proteins called signal recognition particles. This is a signal recognition particle, and they have a groove in the protein structure, and this groove fits or can interact with a specific peptide sequence. So this is an example of sorry, my mic was so loud, of protein-protein interaction, where the signal recognition particle can recognize a protein sequence in the newly formed protein, and then can translocate that protein to the ER. So there's the signal recognition particle, usually that's attached to some sort of filament that will then transport your protein to the ER, where it then gets further modified. So this is Sec61, which can be bound to the ribosome while it's making a protein. And when the N-terminus of your newly made protein comes out of the ribosome, it immediately gets recognized by the Sec61 signal recognition particle. And it will then stop translation. It will tell the ribosome, once it finds this peptide that comes out, it will stop translation and then bring the entire ribosome to the endoplasmic reticulum, where the protein is then, while it's being made, put into the ER. So signal recognition particles or signal peptides are within the protein that's being made. So if a protein is supposed to go to the ER, it has to encode for this little sequence so that it can be translocated there. And there's proteins that recognize those peptide sequences, which will then help in the relocalization. Usually the signal peptide is in the N-terminus of the protein, but one for the mitochondria, for example, can also be in the C terminus. So it just has to be a consensus sequence, but on the amino acid sequence level. Okay, so a sequence that's recognized that is on the amino acid level. Okay, so membrane translocation. The signal recognition particle binds to the signal peptide, that comes out first. So this is your mRNA, your ribosome, and you can see that this is your newly made protein. The N-terminus comes out first, is then bound by this SRP, signal recognition particle, which then moves the whole thing to the ER membrane, and then the ribosome starts translating again, and the protein is actually being secreted into the ER while it's being made. Often the signal peptide can be cleaved off after. So it doesn't always have to be cleaved off, but in a lot of cases, the protein, once it's where it's supposed to go, we don't need the signal recognition peptide anymore. So then it gets cleaved off, which also means your first methionine that you always start your protein with is cleaved off. So these proteins then end up not having a methionine in their fully functional form, but they had it when it was first made. Okay, so if you look at proteins in a specific organism, you might not always find a methionine in their active form, but it was there at one point. Okay, that's good? Okay, and we'll go to another form of proteolytic cleavage. So the cleavage of the signal peptide is what we call proteolytic cleavage. When we cleave a peptide or a protein and we cleave a section off, that's proteolytic cleavage. Sometimes proteolytic cleavage is necessary to activate a protein and make it into its form. And the most well-known one is insulin. So all of you guys have insulin to regulate your sugar metabolism. And insulin isn't actually made in its active form initially. So when insulin is made, it's made as one long peptide here. Okay, this one is called pre-proinsulin. We then have the formation of disulfide bonds here between cysteine residues to form this loop structure that can't be, so these are covalent bonds, disulfide bonds. You've learned about them hopefully in the BOFA section. And the other thing we have taken off is the signal peptide here. After these disulfide bonds are being formed, the C peptide in green, which is part of the initial peptide, gets cleaved off and is being degraded. We don't need that anymore. And this is your active insulin. So your active insulin is 2 peptide strands connected by two disulfide bonds. So here it is, here's your amino acid sequence. You have your N-terminal chain, your C chain, and then your A chain. You have the 2 disulfide bonds being formed, and then you cleave at specific sections here. You don't need to, as always, memorize the sequence, but you should be able to say, okay, it's a loop structure first, disulfide bond next, specifically those 2 here. And then we have the cleavage of the C chain that we don't actually need to have the mature insulin. Okay, so that's how protein cleavage can actually be essential to make a full protein. And we talked about how every protein gets degraded in the end. Some proteins have to be cleaved before they get degraded. So protein cleavage is not always just to get rid of a protein. It can also be to make a functional protein. Okay, so I'll now kind of go, hopefully we'll get out early today because this is the last section for 2288. And then I'll start with 22. The 2280 students are staying on for next topic, but not today. We'll do that the next lecture, I think tomorrow. So I'll go over all the forms of protein regulation, and then we'll have a little bit of new stuff in the very end where we go over protein inhibition. Okay, so the amount of active protein in a cell is regulated at a given time on many different levels. And we've talked about basically all of those levels in the past 6-7 lectures. So you have your DNA is being made into RNA, it's being made into translation. And this looks very simple, but really at every single step here, we have regulation. We have regulation at the transcriptional level, how much RNA is made, how much RNA stays. So the regulation of RNA is also at the point of where it gets degraded. How much RNA is available for translation. Then translation itself is regulated by the different ribosomal binding sites. And then we have the making of the protein. And the proteins are also regulated in how active they are, where they're localized, whether they're properly form, whether pre-insulin is processed into insulin, also a form of regulation. So there's many different layers of regulation. In many cases, the regulation starts at the gene expression. And we can regulate gene expression by preventing or reverse disease. So a lot of researchers are trying to work on regulating gene expression in order to allow for proteins not to be made, or for proteins to be made at a higher level. And there's like small chemotherapeutics that are being used in order to regulate gene expression. So the other time that we use gene expression is when we make proteins in the lab. Okay, we also, when we make a protein in E. coli, we first let E. coli grow so that it's happy, and then when it's like at its happiest, we make it do all the hard work. But if we would do this in the very beginning, the E. coli wouldn't even divide enough to make any protein. So gene regulation is used in therapeutics and in the lab as a tool. Okay, so in humans, we have 21,000 protein coding genes, but we usually have less than 10,000 expressed. And this is a two-dimensional gel of proteins from the same human, but from different tissues. How those 2D gels work is we take all the proteins, we extract all the proteins from the cells, in this case from the human brain and the human liver. We then separate them first by just by their charge, basically. So depending on their amino acid composition, they will be more positively charged or negatively charged, and we can separate them by their charge. After we've done that, we separate them by their size. So here, these are small proteins down here, and these are big proteins up here. So these are acidic proteins, or proteins with a higher acidic amino acid content. These are basic proteins, and then these are big proteins up here, small proteins down here. And if you look at these two gels, first they look like just a bunch of dots, but every dot here represents a protein species. So if I were to do an analysis by mass spectrometry on one of those dots, it will come back as saying, this is your myosin protein. Okay, so every single dot stands for a protein. What you can see here is that we have differences between the brain and the liver. So if you look at this one, there's a lot of a specific protein right here. And then you can see it's not really here. There's not much here. Or if you look up here, the brain doesn't have this one specific protein or this group of proteins, but the liver has a ton of it. So these are likely proteins that are just needed for liver function and not for brain function. So you can imagine that the liver needs proteins that are involved in converting one chemical to another, one toxin into something not toxic. So these are enzymes that are needed for a specific function. And then the brain might need, you know, signal transduction things to transduce signals between one axon to another. So these ones wouldn't be needed in the liver. So this protein level, you can also see big differences here and here compared to here. So these, even though they have the same genome, they don't have the same proteome. Okay, so when we do genome sequencing, it'll only tell us so much about the actual function of the cell. Okay, so where do we regulate? And this is kind of a recap of the past lectures. We regulate at the transcriptional level, initiation, elongation, termination, especially at initiation. We've talked at length about how initiation can be regulated. RNA processing, the editing, capping, splicing, polyadenylation, all of these are regulatory steps. And any step you introduce gives you another layer of regulation. mRNA export, we didn't talk too much about that, but it's another form of regulating how much mRNA is available. This obviously only in eukaryotes. how much mRNA is available in the cytoplasm. So mRNA that's in the nucleus doesn't do anybody any good. So if it's in here, nothing happens to it. It has to go to the cytoplasm to be made into protein. So that's another form of regulation, is how fast or slow do we export a specific mRNA. And then mRNA degradation, if you degrade the mRNA and reduce its stability, there's nothing there to express anymore. And this is something that's the degradation and the rate of decay, something that's getting more research interest now that we have better sequencing method and we know more about mRNAs. So the past few years, the mRNA field and its regulation has gotten a lot of interest. And if you ever hear about siRNAs, we can actively regulate a cell by putting small RNAs into it that interfere with degradation. So there's lots of new technologies out there about mRNA degradation and how to regulate it. Okay, protein translation, the rate of initiation, elongation, termination, same thing. So if you don't make the protein or you make it very slowly, that also changes how much protein is being made. One thing that's interesting is the amino acid composition of a protein also determines how well it's made. So if you have a protein with a lot of rare amino acids like tryptophan, it will be made slower because the cell has less tRNA trip around because it's a rare amino acid. So that will slow the ribosome down at any given, like when it waits for the trip codon to come in. And that would also change the rate of protein translation. Okay, finally, protein modification. The post-translational modifications that we talked about, they are a huge, they have a huge impact on the activity of a protein and its function. We guys saw the P53 protein and how many different signals it can take in order to change its function. So some proteins, even though they're made and properly folded and done, they're not active at all unless they get a phosphorylation. Okay, so only when they're activated by phosphorylation will they start their function in whatever signal transduction or whatever they need it for. Same acetylation, insulin cleavage. Insulin is not active as pre-insulin. Okay, pre-insulin just floats around until it's needed, and that gives us a quick response. So when you have this regulation on the post-translational level, the advantage you have with that is that you don't have to wait from the making. If you comparing post-translational modification as a regulation tool and transcriptional regulation. When you regulate at on the transcriptional level, you still have to make the whole protein and process it, right? You have to go through making the RNA, exporting, making the protein, activating it. When you already have your protein level here and you just turn it on and off by a post-translational modification, that's a very quick on and off switch that the cell can use really quickly to adapt to new environmental challenges, which is why we think insulin and pre-insulin are modified the way they are, because you can have a lot of inactive pre-insulin in your bloodstream that can be quickly converted into the active form, right? So you don't have to wait a long time to react to a sugar shock, which is essential for the cells, because otherwise you get in trouble if your sugar levels get too high. Okay, and then finally, protein inhibition and degradation. And this, we haven't talked about protein inhibition much at all. So that's what we'll talk about in these final, 10, 15 minutes. So proteins can be activated by post-translational modifications, or they can be inhibited by also inactivation, but also by small molecule inhibitors. So this is how many drugs work, and we'll talk about aspirin, because that's something probably everybody has at some point in their life taken and never thought about how it actually works. Aspirin is a small molecule that inhibits a protein called cyclooxygenase, or short COX. So COX is inhibited by arachidonic acid, aspirin, and it can, aspirin inhibits COX. What COX does, it converts arachidonic acid, and it has two functions. There's two different COX proteins, COX-1 and COX-2. COX-1 has the housekeeping function, and it's mostly expressed in the lining of your stomach. Okay, that's COX-1. COX-2 looks very similar, but it's not exactly the same. We'll look at the difference in a second. And COX-2 is not housekeeping, meaning it's inducible and it's only expressed when needed. And it reduces inflammation and neoplasia. So COX-2 and COX-1 are both being inhibited by NSAIDs and by aspirin, even though aspirin is not technically an NSAID. Okay. There are three types of COX inhibitors, and you've learned about the different. Have you learned about inhibition with Dr. Bofa? Not sure? Not really? Okay, so you don't need to learn the types of inhibition for my class. If you haven't learned it for Bofa, then you don't need to remember this. I just want you to remember that there's different types of inhibition, and these are the different NSAIDs have different versions or different forms on how they inhibit your COX proteins. So this is aspirin and ibuprofen, and they work slightly different. The way how ibuprofen works, it inhibits, it binds to the active site of COX, but it can go out again. Okay, so it's a reversible inhibitor that can go in and out of a protein. And reversible is usually non-covalent. Okay, so it can go in and out and competes for the substrates on the active site, but it can stop inhibition at any time. Whereas aspirin binds irreversibly. So once it's in the COX protein, it stays there. And if you want active COX protein, you just have to make a new one. Okay. Okay, so COX-1 and COX-2 are both being inhibited by aspirin. And this is aspirin here or here. You can make aspirin in any biochemistry or high school lab. So Bayer, the company that invented it, had a patent on it for decades, allowing nobody else to make it. After they made it, after the patent ran out, the price dropped dramatically because it's super cheap and easy to make. So you can and I've done that when I was in high school. We made aspirin. I wouldn't recommend eating anything that you make in a lab. I would, trust the pharmaceutical companies and not myself. But in the lab, you just take salicylic acid and acetate anhydride, and that makes you aspirin. And it's really quick, easy, and cheap to make, which makes it a great medication because it's something that's cheap, but you can sell it for a lot of money. So pharmaceutical companies obviously like this. And what it does is acetylates a serine. Okay, so it modifies the COX protein by putting an acetyl group on a serine in the active site of the COX protein. Okay, so aspirin binds to COX-1, and if you look back here, COX-1 is the one that has the housekeeping function and helps in the protection of your stomach lining from the acid. So your stomach in your body has a lining that protects it from the stomach acid, which is why taking a lot of aspirin can lead to stomach ulcers, because you're losing the stomach lining, and then you have your stomach cells, the surface cells exposed to the acid, leading to stomach ulcers. That's the reason why aspirin does this. The others, ibuprofen, they don't target COX-1. They target just COX-2 because the active site of these enzymes looks slightly different. Okay, so aspirin will block both COX-1 and COX-2, but preferentially COX-1. And it's an irreversible binding, and it acetylates by acetylating a specific residue of COX. Okay, so they were like the pharmaceutical companies were thinking, okay, so if you have a COX-1 inhibitor and it affects the stomach lining, maybe you don't want that. Maybe you want a more efficient COX-2 inhibitor and not inhibit COX-1, because COX-2 is the one that does the inflammation. Okay, COX-2 promotes the inflammatory response. If you want to reduce inflammation and reduce the nervous system reacting to inflammation and headaches and whatever other pains, you only want to block COX-2. So they looked at the crystal structure of COX-1 and COX-2, and it's really a very small difference. So these are two isoforms of the same protein. They're not splicing variants. They're encoded by two different genes. And the major difference is really in the COX-2 active site, you have a valine here, whereas in the COX-1 active site, you have an isoleucine in purple. And you can see the isoleucine is larger than the valine. So what you would want is to have an inhibitor that would work with COX-2, but not with COX-1. So you just need to design an inhibitor that is big enough here so that it can't, COX-1 would clash with it. So COX-1 will not allow it in the active site. And, but COX-2 would still allow it to bind. So they designed this inhibitor and thought, this is great. It went through clinical trials, all was, fine. And it was called a super aspirin because it was really efficient at just inhibiting COX-2. Which it was really good, but we didn't realize that binding to just COX-2 and binding so efficiently gives us problems that we didn't quite see coming. So it was used a lot, and you know how over-counted medicines are just being kind of used like candy in a lot of cases. And then FDA analysts saw an increase in heart deaths and heart-related disease. And it was then estimated that Vioxx, which is the super aspirin that was generated, caused between 88,000 and 139,000 heart attacks, 30 to 40% of which were probably fatal in the 5 years that it was on the market. So sometimes designing something that's better or a greater inhibitor to a certain protein doesn't give you the result that you thought it should give you. And it took a while to associate these heart attacks with the Vioxx because it's an over-the-counter medicine. So do you really know what medicine the patient that came in with a heart attack took before they had a heart attack? You usually don't. And do you know which kind of pain med are they using? Are they using ibuprofen or Celebrex or Vioxx or aspirin? We often have no knowledge about that sort of history. So moral of the story is, the clinical trials don't always see everything. But on the other hand, aspirin and ibuprofen and all these NSAIDs have added a lot of quality of life to a lot of people and have helped a lot of people. So since then, now on all the ibuprofen packages, all the NSAIDs have warning labels on them. Warning, this can cause a heart attack. Okay, so take them with care, basically. Which, with that, I am done with the content for 2280, and I will let everybody go home and enjoy their afternoon early. Hopefully you don't have too many more classes. Thank you. I do have a video for those who don't have anything else to watch or anything else to do, which is a 1960s. Hold on. Oh, come on. Hold on. Okay, I can't even start it.
Uracil yellow for admin. If there is a message, there must be a way to translate that message. That's the job of the ribosome and of the transfer RNA. Ribosome is composed of a large and of a small subunit.
Okay, so for those of you guys bored, this is a video that Stanford scientists made in the 1970s, and I don't know what they smoked. They certainly had a lot of fun.
The small subunits.
explaining
with the aid
translation.
of
So
an
if you ever want to
initiation
Google
factor,
Stanford
captures
translation
the message.
1970s, it's super
Then
funny.
The first
right.
transfer RNA
Have a great
carrying
day.
its cognate amino acid is brought to the ribosome message complex by a second dancing initiation factor. This requires energy, and that's represented by a puff of smoke. Next, the large ribosomal subunit tumbles into place, and then the process of bringing each amino acid to the ribosome message complex is accomplished by the T factor and its GTP cohort. And so, in the order prescribed by the balloon colors in the message chain, each amino acid is brought to the assembly site to be added to the growing chain by the peptidyl synthetase. Next, in an energy requiring step, the message RNA, tRNA complex, is shifted so as to bring the next message unit into the ribosome to allow the process to repeat itself. At the end, the terminator factor, seeing the termination signal, cleaves the completed protein from the last tRNA, releasing it from the ribosome. So that the ribosome can do its job again, the two subunits are split apart and separated from the messenger by the ribosome releasing factor. My diagram is of necessity static, but protein synthesis is a dynamic process. This movie tries to bring those dynamic interactions to life.
Twas Brillig and the slithy 30S ribosome did gyre and gimble in the wabe. 30S ribosome. All mimsy was mRNA, that colored message unit array. mRNA. Bound they were in the glorious sun by initiator factor 1.
Initiator factor 2.
Initiator factor 2 went searching for tRNA, who bore the flaccid amino acid.
tRNA! GTP! Woo!
Then, as the uffish bonds were formed, the 50S ribosome untamed came whiffling through the tulgey grass and burbled as it came.
50S ribosome! 50S ribosome! 50S ribosome!
factor plus GTP gathered in tRNA.
tRNA! Woo! Wow!
Gathered in tRNA and came galumphing back.
Oh, hydrogen bond!
Amino acids both inside, alas, the peptide bond was formed.
Peptide bond!
And as the index step took place, the message units shift a space.
tRNA! Acidyl synthetase peptide bond. Index! tRNA!
Each tRNA approached the site bearing its amino acid load, whose sequence was determined by the mRNA message unit magic code.
Whoa, whoa,